Hi girls, final video in the aerobic cellular respiration set. This is the electron transport chain. Guess what? There's another wrap. A few years ago, a girl memorized at least the chorus of this and delighted on singing it with me, or singing it to me at the beginning of every lesson. I set you that challenge. Let's see how you go. All right, so this is the final stage. This is arguably probably the most important part of aerobic cellular respiration. So prior to this, we haven't talked about oxygen at all. So we know that oxygen needs to come into cellular respiration, but we haven't used it yet. So this is where the oxygen comes into play. We've also made a little bit of ATP. Glycolysis made 2 ATP. Krebs cycle made 2 ATP. But this is where we're going to chunk out most of our energy. So there's not much point in doing aerobic cellular respiration unless you can make a lot of energy, because it takes a lot of effort to go through to this. So the point of the electron transport chain is to move protons and electrons, across a membrane, and we want to generate ATP. Now, the thing with the electron transport chain is we want to make lots and lots and lots of ATP. You must have oxygen for this to work. So we said ages ago that we talked about aerobic respiration, and aerobic respiration made a lot more energy, but it was a lot slower. So this is where we're getting our energy from, all that oxygen. And this one occurs in the inner mitochondrial membrane, or the cristae. Okay, so you can see here is my picture of my little mitochondrion down the bottom. You can see the folds here of the cristae, and here is your outer mitochondrial membrane. Let's zoom in now. Over here is my inner mitochondrial membrane, or the cristae, and there are a bunch of these proteins embedded in that membrane, including this guy, ATP synthase. Now, the whole point of these is that they form, can you see this? And I'm just going to make a different color just to make sure that we can see that. Or not. Okay, can you see how there's this little line that's following all the way through? That is your chain. So the electron transport chain is about using electrons and protons all the way along the chain to make lots of ATP. Again, we don't need to know the inner workings of the electron transport chain. We're talking second, third year uni, possibly later. So this is where our coenzymes come into play. So we've made 8 NADH. Where did we get them from? Where have I come up with this magic number 8? So I've got 2 from glycolysis and 6 from Krebs. Okay, so that's where that's come from. We've also got this 2-FADH2. Again, way off the study design. You don't need to know about them, but they've come from the Krebs cycle as well. So we get those coenzymes from glycolysis and the Krebs cycle, and we use them to pump the protons into the intermembrane space. I'm going to show you this in an animation in a second. When you pump them with active transport, we're moving against the concentration gradient. So you're putting them in a position where there are too many of them on one side. So what we want them to do is they're going to fall back down. Okay, they're going to fall back down. Now I'm going to change this number because I've realized that I didn't have it changed on here. It should be 32 or 34 ATP. This is contentious, this number that we're going to talk about, just because different books say different things. The biggest thing with this is you need to know that it's lots and lots and lots of ATP. As a result of this, those protons have to go somewhere. So a proton, for the chemists in there, is an H+. Okay, so what are you going to do with a bunch of hydrogens when you've only got oxygen? So what you do is you get your oxygen, you add in the hydrogen, and all of a sudden, you've got water. So this is where the water from cellular respiration happens. Here's another example of why it's a chain. Can you see how you're using NADH, this other one called FADH2, we're dropping off the H's. The H's are being dropped off, they're pushing them up. We've got a high concentration of H+ ions over here, and what they do is they fall back down through this one here called ATP synthase. What do you reckon it does? It synthesizes ATP. It's also an enzyme. Okay, so here's my little animation just to show you what's going to happen. So we're going to start over here with the purple NADH. So we start over here. You can see that we have a whole bunch of hydrogen ions inside the matrix. Okay, so we're inside the matrix here. Now this NADH is going to move along through the chain, dropping off its hydrogen ions and using the energy, to pump those hydrogen ions through into this inner membrane space. Once they're all in this inner membrane space, you can see you've got a high concentration of H+ in here. What are they going to want to do? They're going to want to diffuse back down. So they're diffusing back down through ATP synthase all the time. This ATP synthase is using the power of that pump, of that diffusion, to create, it says 26 to 28. I'm going to change that. It's going to be 32 or 34 ATP. Okay, and that is the general gist of what's happening. Now, the problem is you've got all these leftover hydrogen ions. What are you going to do with them? You need to make sure that you keep your concentration of hydrogen ions. So we want the concentration of hydrogen ions to be low. So what are we going to do with them? We're going to use them with the oxygen that we've brought into the cell and create water. Okay, so it is extension. The hydrogen ions are coming from the NADH and the FADH2. So here is a big summary picture of everything that's happening. I'm just going to skip through them just so we can go on to this bit here. So this is the ETC. What do I need to remember? You need to know that it happens on the cristae or on that inner mitochondrial membrane. You need to know what you put in. You put in oxygen to the ETC. You get out water. Again, that water is not what we need. What we actually want from this ETC, this is what we want. What we want is the ATP. The water is just a waste. And you can see that water. Do some heavy breathing on your hand, touch your hand, and if it's moist, then what you're seeing is the water that you're not needing anymore. So we can breathe out a lot of the water that we produce. We also are going to use all of our NADH from glycolysis and Krebs, and we're going to reconvert them back to NAD+. So here we have our unloaded coenzyme again. And here is our loaded coenzyme. So we loaded up the NADH in Krebs and glycolysis, and this is why. We've done it so we can use it for the ETC to make what we want. So what we want out of aerobic cellular respiration is lots and lots of ATP. And that is the end of that video.